Okay, good evening, my dear friends. Regarding of CPGET 2024 Botany free online coaching, our resource person, very huge experience personality, dynamic botany faculty, that is none of the other than Dr. P. Manjula Madam Garu, Assistant Professor, Department of Botany, Food and Nutrition. Madam has clearly explained in previous classes about the plant anatomy and along with ecology, biodiversity. Now, today, Madam is going to explain about the most important topic in the cell biology or cytology. Previous class, Madam has clearly explained about the cell structure, general information, along with cell organelles, important cell organelles, and Golgi apparatus, mitochondria, cytoplasm, and other cell organelles. Madam has clearly explained. So, remaining topic and continuation topic, Madam is going to explain today. Madam, please welcome, hearty welcome, and share your screen. Start your session, Madam. Please welcome.
Okay, thank you, sir. Sir, is it visible?
Yeah, visible, Madam.
Okay, so today's topic is
Madam,
composition of...
possible click on slideshow. Yes, okay.
Okay. So next topic, that is a composition, that is chemical composition of the cell wall. So generally cell wall, it was the first observed and named simply as a wall by Robert Hooke in 1665. So generally cell wall which is present in the plants and plants and it is absent in the animals. So in 1804, Carl Rudolphi and J.H.F. Link proved that cells have the independent cell walls. So a cell wall, it is the structural layer and that surrounds some type of cells and situated outside the cell membrane. So generally cell wall, it is present outside the cell membrane. So it can be tough, flexible and rigid, and which provides the cell with both structural support and also the protection. The main function of the cell wall is protection and also gives structural support, gives rigidity to the cell. So on the basis of chemical composition of the cell wall, so there are three types of cell walls. That is the bacterial cell wall, which is made up of mucopeptides and muramic acid, and fungal cell wall, it is made up of chitin, and plant cell wall, generally it is made up of cellulose. So this is also important points regarding the competitive exam. That is the cell wall of the fungi is made up of, that is chitin. And cell wall of plants, generally cellulose, and bacterial cell wall, it is made up of mucopeptides and muramic acid. So generally bacteria, it is bacteria which constitute the large domain of the prokaryotic organisms. So the study of bacteria, it is known as a bacteriology. It is a branch of microbiology. So bacteria was first discovered by the Anthony van Leeuwenhoek in 1970s. So here again, here two types of cell wall, that is gram positive and gram negative. So here the peptidoglycan is present in the gram positive, whereas here the less amount of peptidoglycan and it is covered with the outer membrane in the gram negative. Okay, so the difference between gram positive and gram negative. So peptidoglycan, it is also known as a murin. It is a polymer which consists of sugars and amino acids that forms a mesh-like layer outside the cell membrane of the most bacteria forming the cell wall. So this sugar component, which consists of the alternating residues of beta-1,4 linked N-acetyl glucosamine and N-acetyl muramic acid. So these subunits, which are related to glucose in their structure, and they are covalently joined one another to form the glycan chains. So types of bacterial cell wall. So based on the structure of the cell wall and their and to their response to the strain, bacteria have been classified into two types. That is a gram positive bacteria and gram negative bacteria. So gram positive cell wall means it is usually thick, homogeneous and composed of mainly peptidoglycans. That is 50 to 90% of peptidoglycans and the dry weight of the cell wall, and contains large amount of teichoic acid. Whereas the gram negative cell wall, it is multi-layered and more complex than the gram positive cell wall. So here the peptidoglycans of the gram-negative bacteria is thin and comprises only 10% only, and less of the cell wall. So outer membrane which lies outside the thin peptidoglycan layer. So most abundant protein, that is bronze lipoprotein, is seen in the gram-negative cell wall. So here bacterial cell wall, again that is the gram-positive and gram-negative. In the gram-positive, generally more amount of peptidoglycans are present. So when the stain with this gram staining, so generally gram positive bacteria which gives violet color or purple color, whereas here the gram negative it gives pink color, the fixation of the gram stain. So crystal violet, that is iodine treatment, and decolorization and counter stain softening. So here generally when we stain gram staining by using this gram staining technique, so gram positive bacteria they gives violet or purple color stain, whereas the gram negative bacteria which gives a pink color. Okay, so generally example for this gram positive is cocci, round shaped bacteria, whereas the gram negative bacteria example is bacilli, that is rod shaped bacteria. So gram negative bacteria is here bacillus. So functions of the bacterial cell wall, so which protect the cells against the osmotic shock and physical damage, and regulation of the substance transport into and out of the cells, and contains supplementary genetic information such as the resistance to the antibiotics, production of toxins, and tolerance to toxic environment. So which take part in the protein synthesis, movement of the cells, and mineral storage of the cells. So these are the functions of the bacterial cell wall. So next cell wall, that is the fungi. So generally fungi, they are the eukaryotes. Nearly all multicellular except yeast are the unicellular and spore-bearing protista that lack the chlorophyll. So generally in the fungi, that is chlorophyll is absent. So generally eukaryotic and heterotrophs are the fungi. So generally two types here, yeast and molds, that is filamentous and multicellular. So fungal cell here, cell wall, which is the cell wall is made up of chitin, that is a polymer of acetylated amino sugar and N-acetyl glucosamine, and glucans, proteins. So glucans and chitin are the components of the primary wall, and proteins are the components of the secondary wall, and other components include chitosan and melanins and lipids. So these are present along with this. So these components also present. So mainly it is made up of fungal cell wall, which is made up of chitin. Along with this chitin, glucans and proteins also present. And in the primary cell wall, glucan and chitin are present, and proteins are the components of the secondary wall, and other components include chitosan, melanins, and lipids also present. So functions of the fungal cell wall, so protects against the osmotic lysis, act as a molecular sieve, contains pigments for the protection, and binding sites for the enzyme. So the outermost surface of this the cell wall, which provides medium between the cell and the environment, and it is the site where antigen and agglutinin gets attached to the substrate, host, and other cells. So next one is the plant cell wall. So the plant cell wall, which is a remarkable structure, so it is provides the most significant difference between the plant cells and other eukaryotic cells. So the cell wall is rigid up to many micrometers in thickness and gives plant cells, it is a very defined shape also. So most cells, they have the outer membrane. So it is none is comparable to the strength through the plant cell wall. So the cell wall is reason for the difference between the plant and animal cell function, because the plant has evolved this rigid structure. So the components of the plant cell wall are mainly here again the plant cell wall is again it is different layer composed of different layers like middle lamella, primary cell wall, secondary cell wall, and tertiary cell wall. So first one is the middle lamella. So it is present between the two adjacent cells. So two cells are connected through this middle lamella. So it is situated outside the primary cell wall and is made up of calcium and magnesium pectate. So generally middle lamella, which is made up of calcium and magnesium pectate, and it acts as the cement which holds the adjacent cells. So it acts as a glue or cement between the two cells, adjacent cells. Next one is the primary cell wall. So it is formed after the middle lamella, and it is a thin, flexible, and extensible layer. So it is capable of growth and expansion. So the backbone of the primary cell wall is formed by the cellulose fibrils, and the matrix is composed of hemicellulose, pectin compounds, lipid, and structural proteins. So this is the structure of the primary cell wall. So it contains middle lamellum, and here this one is a primary cell wall. Here hemicellulose, pectin, and cellulose, microfibrils, and soluble proteins are present in the primary cell wall. So next one is the secondary cell wall. So it is extremely rigid and provides strength, and it is not found in all cell types. So generally secondary cell wall which may be present in some parts of this cell only. So it consists of three layers known as the S1, S2 and S3 layers. So S1 is the outer layer, S2 is the middle, and S3 is the inner. So it is composed of the cellulose, hemicellulose and lignin, secondary cell wall. So next one is the tertiary cell wall. So tertiary cell wall, it is deposited in few cells. So it is considered to be the dry residue of the protoplast. So besides the cellulose and hemicellulose, here the xylan is also present in the tertiary cell wall. Next one is the plasmadesmata. That is they are the protoplasmic strands that connects the protoplast of the neighboring cells. And the diameter is 40 to 50 nanometers. So two adjacent cells, they are connected through this plasma. So small openings which are present at the adjacent cell walls. So through this, here the connection between the two adjacent cells will take place. That is the transport of the any material between two cells. So next one is the pits. So in cell wall, that is a perforations are found in the form of the simple bordered pits. So simple pits are thin-walled spots which are formed at certain points. So where the deposition of the layers of the secondary wall does not take place. So bordered pits are the pits. So two types of pits, that is the simple pits and bordered pits. So simple pits are thin-walled spots, whereas the bordered pits which are partly covered by the innermost layer of the secondary wall as found in the dead tracheids and vessels. So this is the bordered pit. So the structure of the bordered pit, it contains torus, pit aperture, and next one is the pit border and pit membrane. So this is the structure of the bordered pit. So next one is the composition of the cell wall. So the cell wall is mainly composed of, so what are the chemicals which are present in the cell wall? Okay, in last that is the what are the layers and different components, that is outer layer, so are present in the cell wall. So next one is the composition of the cell wall. That is the cell wall is made up of different types of chemicals like cellulose, pectin. So the cell wall is mainly composed of carbohydrate materials, and the major components of the cell wall are cellulose, pectins, hemicellulose, proteins, and phenolics. So cellulose, so it provides shape and strength to the cell wall, and it composes of 20 to 30% of the dry weight of this primary wall, and accounts 40 to 90% of the dry weight of the secondary wall here. Okay. So pectins, so they are the group of polysaccharides which are rich in galactouronic acid and rhamnose, arabinose, and galactose. So pectins are present in high concentrations in the middle lamella, where they are presumably serve the function of the cementing. So because they are mainly, the main function is cementing the two adjacent cells, pectins. So this is the composition of the cell wall. The cell wall mainly composed of cellulose 10 to 15%, hemicellulose 5 to 15%, water 60%, proteins 1 to 2% only, and lipids also 0.5 to 3%, and pectic substances 2 to 8. So this is the composition of cell wall. So more amount is, that is cellulose, and that is water content, and cellulose is more in concentration. So hemicellulose. So hemicellulose, these are the matrix polysaccharides which build up the variety of different sugars. So the different species and different cell types also. Xylan. So it is also a typically make up the roughly 5% of the primary cell wall and 20% of the secondary cell wall in dicot. So the hemicellulosic polysaccharide is linked with the xylose and arabinose. So proteins. So different varieties of proteins are present in the cell wall, the most of which are linked with the carbohydrate, forming the glycoproteins. That is proteins with carbohydrates forming the glycoproteins, and the cell wall of glycoprotein extensin. Extensin contains the unusable amino acids, that is hydroxyproline, about 40%, so which is generally absent from the protoplast. So these extensins are present in the primary cell walls of the dicots and making up 1 to 10% of the wall. So these are the main components in the cell wall. So what are the differences between this primary and secondary wall? So primary wall generally it is in all the plant cells, primary cell wall is present. Here the secondary cell wall only in mature and non-dividing cells. Position generally inner to the middle lamellum, whereas the inner to the primary. So inner to the primary cell wall, here the secondary cell wall is present. So nature is, it is elastic and thinner. Primary cell wall is elastic and thinner, whereas the secondary cell wall, which is inelastic, rigid and thicker. Next, nature of the growth is into suspension. And here accretional, that is addition of this deposition of the material will take place. So here pits absent and here present, and additional material here absent and present lignin, suberin and cutin. So based on the cell type also, here the secondary material may be varies. So amount of cellulose is low in primary cell wall and high in secondary cell wall. So extensibility, that is present in primary cell wall and generally absent. So arrangement of the fibrils, wavy and loosely arranged in the primary cell wall, and these fibrils are arranged closely, straight and parallelly arranged. So hydration more, that is 60% of water, here less, that is 30 to 40%. Secondary cell wall contains less, that is 30 to 40% only. So functions of the cell wall, they determine the morphology, growth and development of the cells, and they protect the protoplasm from the invasion by the viral, bacterial and fungal pathogens. So main function is protection, and they also rigid structure and thus helps in the plant in withstanding the gravitational forces. And they are also involved in the transport of materials and metabolites into and out of the cell due to presence of this plasma desmata. So they withstand the turgor pressure, so which develop within the cells due to the high osmotic pressure. So these are the functions of the cell wall. So eukaryotic cell walls, that is composed of polysaccharides and polymer. So chitin of and cellulose joined by beta 1,4 linkage. So in fungi, the example fungal cell walls, algae and water molds, slime molds, etc. So fungal cell wall, it consists of chitin and polysaccharides. Matrix of the three components, that is chitin, glucans and proteins.